We're going to hear now from the scientific strategy co-chairs and a colleague, I believe, as well, who can update us on the work that they've been doing. We'll see the three co-chairs that have been working with this group over the year, and today we're going to hear, I believe, from Adam Butterworth, who's at the University of Cambridge, Håkan Håkansson from Children's Hospital of Philadelphia, as well as Patrick Slayman, who is also at CHOP in the U.S. And so I've taken a peek through the slides, and again, we'll turn it over. I'm not sure if Adam or Håkan is getting us started, but to hear about the different proposals and ideas that you have already been working on, as well as those that you're continuing to think about.
Yeah, thank you, Laura. So I was actually going to start, and then Patrick will be in the middle, and Adam will close. As Laura said, we're going to update on progress within the genomics working group on the scientific strategy and sort of, how these projects have advanced. Patrick will be presenting early analysis on polygenic risk score. This is a pilot that sort of crosses multiple cohorts. And then Adam will close on the next steps and followed by questions and answers. Next slide. And probably the next one. So we have had several meetings with ongoing efforts sort of focused on developing the proper infrastructures that would allow for cohorts to collaborate and share data across the continent, and not just data, but expertise and other elements, with the goal of sort of creating a collaborative, vibrant scientific initiative that sort of encompasses the large-scale programs that we have here at Penn. And as Geoff indicated in the beginning, there are 97 cohorts that have signed up for the IHCC with over 50 million participants. So there is enormous potential here going forward with the deliverables. And our goal is really focused on sort of, focusing at least the early initiatives to be integral to clinical care of the populations that we serve. So you heard from the previous groups, Group A and C, before in terms of the infrastructure and where we stand. So the data atlas, we have a working model sort of essentially in place. This is just about to be released, as you know. The consortium charter is similarly essentially good to go, and there will be an MOU which would be subsequently signed off on as a continuum of that. And there are a number of efforts that have been focused on fundraising, and we heard the willingness from Francis Collins this morning from the NIH and the Wellcome Trust, who have already contributed funds to these programs. So we sort of reference this as the phase 1.5 for the implementation of this initial pilot. So it was in late December that we sort of got the word that there was some potential funding coming from the NIH to execute a pilot project across multiple different cohorts. And the goal was to try to get sort of a proof of concept demonstration in place about the feasibility of doing this. And obviously, without having the charter, without having the MOU, there were obviously several sort of discussions. So it took a bit to sort of, get this moving, and we only had 4 months up to this date. So the cohorts that actually ended up participating in the risk score, not all of them made it in part because of the COVID situation, and there's some delay, but they are still going to advance the data, so there will be more comprehensive analysis. But we got multiple cohorts, data from multiple cohorts, and this mechanism was worked in such a way that the polygenic risk scores for 4 or 5 different traits, including diabetes and asthma, height, and BMI, they were generated and then validated, and then the algorithm was submitted to the sites that were actively participating, and they, generated summary statistic data that was subsequently centralized and analyzed. So this was sort of a federated model, if you will. And Patrick Slayman led this effort and will be presenting on it shortly. So this was sort of a fairly lean and mean structure, but with some funding being provided to each side who participated to allow this to happen and sort of, leverage these existing resources of data that could generate summary statistic in a very short period of time. And next slide. And so this was certainly in keeping with one of the key principles of the IHCC consortium, to maximize the bang for the buck, if you will, and try to generate the most impactful discoveries for comparatively little investment and as rapidly as possible, because the timeline was certainly very expedited. And the polygenic risk score pilot program is really the first example of a network-wide approach that we will be showing off shortly. And so the pilot projects that we dealt with last year after the meeting in Iceland, there were multiple projects, and I will go through a few of them in a couple of slides. But we sort of selected a couple of themes that we were interested in advancing if there was a successful fundraising effort to fund them going forward, the polygenic risk score being one of them. Another was assessment of risk in sort of, underserved populations. And the third one was on a human sort of loss of function knockout variants. And the fourth on pharmacogenomics. And we also came up with a framework to prioritize projects sort of with the notion that here would be those sort of 6 key elements that we would use to judge future projects. And as you all know, the program is now open for submission of additional projects in addition to the ones that we dealt with last year. And the key criteria for to prioritize would be to show innovation for these existing resources in these proposals, have a broad scope across multiple cohorts, quick win across sort of a short period of time, likelihood to elicit enthusiasm by funders, and that the research team and associates would obviously have to have the proper expertise and resources needed to implement the project. And a particular emphasis also on the opportunity for career development of junior researchers and junior faculty. And so, so for the implementation of priorities, we have been sort of, keeping this in mind in terms of how we prioritize and really to sort of, focus on cohort enhancement that would allow countries and regions that have large cohorts and other types of data, associated data, but may have limited resources to advance their genomic progress. And example of this would be projects that identify resources that would be needed to convert samples into data, and particularly with focus on diversity to bridge the diversity gap that we have in multiple ethnic groups, and genomics with focus on low and middle income sort of families. And also prioritize projects that would improve our understanding of the causality of disease. This, of course, is critically important, and examples would include proposal that sort of would focus on developing a robust diagnostic tools and technologies that would allow us to identify high-risk patient populations and propose subsequently innovative analysis across multiple diversity cohorts and groups. And a third element would be focus on, for priorities, focus on projects that sort of assigned or designed to improve our understanding of the variability in response to therapy. And this would be sort of some novel pharmacogenomic associations across cohorts, and obviously we focus on diversity populations as a highest priority. And the fourth element, additional enhancement to existing cohorts and data sets, sort of, if there was ability and funds available to conduct multi-omics studies, sort of, from different platform and implementations, and sort of, take in more fancy digital health devices and other information that sort of, could help similarly with phenotypic gap filling, and again with emphasis on diversity populations. And so I mentioned from the summary meeting from last year, one of the key highlights that we wanted to focus on with respect to implementation was genetic risk factors in low-income family. And this obviously is self-explanatory in terms of how important this would be to address the diversity gap that we have currently. As we mentioned, and IHCC would really be in a robust position to address this and genomically characterize populations ideally if they were well phenotyped. And the considerations that were given was to consider sort of a low density whole genome sequencing where we would get ability to understand the genetic variation across these underserved populations, and to consider subsequently generating a customized SNP array that could be utilized at a much lower cost to generate genomic data across these populations. And the loss of function variants was another big theme, as I mentioned, and the objective here would be to sort of really to create a database or a catalog of high confident knockout or loss of function variants that exist from exome and genomic data. And such a database of loss of function variants would obviously be extremely powerful to identify mechanisms for various sort of phenotypic traits we have, and the ability to conduct potential clinical trials, drug trials, to sort of resemble the impact of a loss of function variant if the loss of function variant was beneficial, or the opposite if not. And this would be also an opportunity, at least for some cohorts, to potentially establish a framework where we can reach out to patients who have and homozygous to allow for additional research and clinical trials potentially to be done. And next slide, we maybe, this is sort of, the summary of the themes that came out of last year's meeting that we have been working on to try to advance as much as possible in multiple meetings, discussions with vendors of these various technologies, but cross-cohort analysis of millions of people is obviously something that is would be very unique to the IHCC. And particularly with focus on rare variants and diversity populations. Phenotype harmonizations, obviously, there's a lot of work with the implementation of that, which is taking place. And GWAS and CNV analysis on existing data, obviously, is a low-hanging fruit. And the COVID research, for example, comes handy into place, where we have large cohorts that are genotyped or sequenced, where we can take that trait of somebody who's positive versus negative following test and run analysis. Exome, genome, similarly, rare and orphan disease, most people are now sort of, moving over to whole genome analysis. And obviously for environmental considerations, epigenetic analysis would obviously be of extended value. pQTLs, there are multiple platforms, SomaScan only being one of them, would similarly be exciting to complement existing omics. And there are various lipid and inflammatory sort of panels that are offered at a very low price that can be run. We had some experience with the scale project, but there are several other ones that similarly could be implemented. So integrative omics phenotype analysis, broad-based across cohorts, would obviously advance the science and also, allow for high impact publications and hopefully interest in third parties' willingness to fund. And this is something that we can, all advance on existing data. And I think the next slide is maybe my last one or close to last one. So we had a recent acquaintance and discussions with a group of scientists and funders and investigators for the Davos Alzheimer Consortium. So this is a global consortium for Alzheimer patients, really focused on discovery, translation, and clinical trial development, and they are bringing together multiple cohorts all over the world, integrating pharma. This is led by George Radenberg, Elias Zerhouni, and Drew Holzapfel, and they are actually going to be presenting on this program tomorrow. And with the army of people that we have in the IHCC, obviously there's a great match with our focus, interest, and initiative. So we've already launched a search for dementia patients within our program to look for a number of cases with Alzheimer's disease, age, range, and diversity, whether GWAS data would be available for, say, polygenic risk score or other types of analysis, and if sequencing data, whole genome exomes or panels would be available, as well as DNA samples and other sample types, such as plasma, serum cells that could be developed into iPS cells and so on. So this is an exciting new initiative, actually initiated by Francis Collins, and the management of IHCC and the Genomics Work Group have sort of taken these first meetings, but this will be a consortium-wide initiative that looks very exciting. So with that, I'm going to introduce Patrick Slayman. Patrick is a statistical geneticist here at CAG and associate director of CAG, and he's going to take you through the analysis that we've done for that initial pilot project for the polygenic risk score. So, Patrick.
Thanks, Håkon. As Håkon said, we're going to present some pilot data around the PRS project. So we selected 4 phenotypes in the end to go forwards with, 2 that were common traits and 2 complex diseases. And really the selection criteria for these was that they would be relatively common and therefore likely to be represented within the site's biobanks. But also, and most importantly, we needed to get this done quickly, and so we needed the meta-analysis data to be publicly available, i.e. we don't have to go through dbGaP requests, which take a little bit of time to get the summary stats. And we also wanted to prioritize studies where there were good, large meta-analyses and preferably transethnic. And so we landed on 4 traits for this initial pilot, BMI, and we were looking both at the quantitative trait and also the dichotomized values at the extremes, both 95 and 99%. Blood pressure, systolic and diastolic, again, we were looking at quantitative traits and we looked at the extremes. And then type 2 diabetes and asthma. And I've listed here, although I'll go through the study design a little bit more on the next slide. I've listed the GWASs that we use for the summary stats. Yeah, there we go. So the BMI, we have the multi-ethnic was from the Giant Consortium Nature paper, and then blood pressure, they're all listed there. European, we had Nature Genetics from last year, and then non-European publication for type 2, we had an Asian, Hispanic, and the European cohort. And for asthma, we had multi-ancestry and the Kappa African American. So next slide, please. So the experimental design, there's obviously a few different ways that you can go about generating PRS scores. We opted to use LD-PRED. primarily, it's not the easiest to use, and that's a lesson learned. There are much more simple techniques such as the PNT, pruning of thresholding, but LD-Pred was attractive for two reasons. One, it takes LD into account, and hence we thought it would actually perform better in a multi-ethnic study. And the other is that it's been shown to outperform PNT, and so we wanted to try and get the best scores that we could. So the overall design was that there's three steps to generating a PRS and LD-Pred. You have to go through a coordination step where you take the summary stats from a previous study and you map those onto your data. You generate LD maps from those. You then use a Gibbs sampler to generate the weights. And then those weights are what's used to generate the scores. So we did the first two steps here at CHOP, and we disseminated the weight files for the sites to generate their scores locally. So in the first, second step, we used a number of different LD files from different populations. We used an African American, Hispanic, East Asian data, European, and then a transethnic, which included all of the above. The real hypothesis that we were trying to test here was whether or not, specifically for populations that don't have good GWASs, whether a transethnic analysis would perform well compared to the population-specific ones. So there's a little bit more detail here. For example, the type 2. So for the Asian data is in the second column here. We had, you start off with your meta-analysis for Asians, Hispanics, Europeans, and the transethnic. In this case, it was a combination of these three. These are mapped to 2,500 imputed CHOP samples that are Asian descent, Hispanic, etc., so that we can work out the LDs. And then using the Gibbs sampler at 8 different fraction of causal variants. So you start off with one, assuming that all variants are contributing equally as causal variants to the PRS, and then you step that down to a fraction of 0.001 and generate scores for all of those. Once you've combined the different fractions with the different LD files, we end up with 40 different weight files that we distributed to the sites. The sites would then run these weight files against their locally imputed data. And I have to say here, so we ended up restricting the SNP content to the HapMap3 data. So they fluctuate around 1 million SNPs. The transethnic had the most at 4.5 million, and the Asian had the least at 174,000. And so the reason we did this was to try and get as good representation out of the box with the genotypes that people would have at their local sites. I have to say, starting off, we weren't even sure what platforms people had genotyped their samples on. We weren't sure who had imputed data versus not. So, these are all things that I'm sure will become easier as a consortium sort of progresses. But as a pilot, we really wanted to make it as simple as possible. And even with using the HapMap3 data, which is a relatively conservative set of common variants, we still had to tweak the SNP content files, these weight files, for different sites. And so the sites would generate their scores. They'd then look at the R-squareds and select the best performing R-squareds for the population-specific data and for the transethnic data, and then generate ROC curves for both of those and provide us with the results. And so we got around not having data sharing agreements and not been able to share individual level data by doing this sort of a process. In this case, all the sites have to do is share have to run the scores and share the results with us. Next slide, please. And so we ended up having these sites participate. ELSA Brazil, the Norwegian Mother and Child Cohort Study, MOBA, CHOP, the two nurses' health studies, 1 and 2, the Shanghai Men and Women study, the UK blood donor cohorts. They provided some data. Not every site provided both BMI and type 2, and I'll go through those in the coming slides. There was also strong interest from 23andMe, the East London Genes and Health, and the Sun and Genome Project. But for purposes largely related to COVID and shutdowns, for example, 23andMe weren't able to get the approvals in time through their internal processes. East London, so the East London group was interested in joining. However, we don't have a large population of South Asians at CHOP, and therefore we weren't able to generate an LD population for South Asians. But these are all things that we can take care of going forward. And the East London group have actually volunteered to assist us with that. Next slide, please. And so we generated the scores, and as you're all aware, I'm sure that not all scores perform equally as well. So the BMI score performed pretty well. We're getting values around 0.73, 0.74. The blood pressure here is on the right. So we looked at European and African American separately. Those are our two largest groups at CHOP. We have systolic and diastolic data. These data were restricted to, because we have longitudinal measures, and as I'm sure most of other sites do as well. So we looked at the measure from the highest age of the participants, and we restricted those to data from kids with age 16 or older. We got some pretty good scores for the African American systolic at 0.74. And then for the asthma performed the least well, actually. So the summary stats that we used were from the recent meta-analysis by Nikolai et al. They had 24,000 asthma cases and 120,000 controls, and they were transethnic. And then we also looked at the CAPA study, which is from Kathleen Barnes's group, looking at 7,000 African and Afro-Caribbean cases versus 7,600 controls. The results were so for our so the phenotype definition from CHOP, we used an algorithm that was developed by the eMERGE consortium by us through the eMERGE consortium, which is pretty detailed. And we still ended up getting AUCs around less than 0.6 at 0.57 for the Europeans and 0.53 for the African Americans. So, not hugely predictive. We then stepped it up in ages, considering the Nikolai meta-analysis would largely be, including adults. The scores did improve somewhat. So if you look at asthmatics over the age of 8, the results improve slightly from 0.579 to 0.584, and they step up a little if you look at over 12s to 0.59. As it turns out, given the COVID situation, most sites didn't have the full capacity to run all 4 scores anyway, and so we ended up prioritizing BMI and type 2 over the blood pressure. And asthma, as the previous 2 are probably an easier phenotype to define, and especially the BMI is present in large numbers. Next slide, please. And so these are some of the results that we got. So looking at the Shanghai health study, so we have the men's health in this column, women's health here, and then the combined in this column. We have the different fractions of causal variance from infinity 1 and then 0.1, etc., stepping it down. These actually should be below these. But these are the fractions of causal variance. And then these are the population-specific data. So the closest matching population LD file would obviously be the Asians for the Shanghai study. And here is a transethnic comparison. And as you can see, in all 3 of the cohorts, the men, the women, and the combined, the transethnic score actually performs better than the population-specific Asian score, which was quite nice to see. So looking at the 0.3 fraction for the men's health study, we have sorry, for the men's is here. So it's 0.76 versus 0.47 for the Asian. Although, so now here you have to consider the fraction. So the actual, as we had said before, the transethnic score includes more SNPs, and so when you step down, when you look at a lower fraction of transethnic variants, they may include the same number of variants as a higher fraction for the Asian-specific population. So 0.72 versus 0.76. Same pattern for the women's study. The transethnic outperformed the population specific at 0.73, and the combined show the same pattern. Next slide, please. The Norwegians, the Shanghai study did not provide type 2 data. The Norwegian mother and child cohort, here we had 50,000 individuals in the analysis with a very decent number of cases for the BMI analysis, 49,000 with 500 cases in the 99th percentile, 47,000 with 2,500 cases in the 95th percentile. The type 2, less so, there were 37 cases out of with 26,000 controls. Nevertheless, in this case, it was actually the European weight file that outperformed the transethnic. I don't have the transethnic data, but the best results that they achieved were at both 95 and 99%, the 0.7 and 0.756 for the BMIs. And the type 2, pretty good at 0.64, considering there were only 37 cases involved. Next slide, please. In terms of the Brazilian, so this is our most challenging cohort, 3-way admixed, of course, with no real primary meta-analysis data to use. And also the numbers were relatively modest. The BMI included 1,781 cases, and the type 2 diabetes, only 310 cases. And yet the transethnic analysis again outperformed all of the three different population specific ones, so African, Hispanic, and European. And the top AUC that we got was 0.645. These are just the number of SNPs used, so it was just under 1 million. Next slide, please. Another large European study. This is the Nurses Health study. Both 1 and 2. One had 121,000 and 2 116,000. BMI was self-reported, and diabetes again self-reported but confirmed by a questionnaire. For the type 2 diabetes, there were some great numbers in the study: 3,500 cases and 1,100 in the second study. The best fraction was 0.1 with an AUC of 0.7. And interestingly, although the European population weights outperformed the transethnic, they were very, very close. So the transethnic was at 0.68 and 0.7 for the European, and these are standardized. And the same sort of pattern for the second study, NHS2. The European only was at 0.65, and the transethnic just below that at 0.646. So next slide, please. Same sort of pattern with the BMI data. Here we had a total of 18,000 and 12,000 with 945 and 669. So it's a good number of cases, well-powered study. Again, the European outperformed the transethnic, but only just. So the best AUC for the BMI at the 99th percentile was 0.729, and for the transethnic was at 0.7269. Same for the 5%, 0.70 versus 0.701. Next slide. And again, same pattern for the, next slide. Next, the same pattern for the NHS2, so 0.735 versus 0.7345 and 0.72 versus 0.718. Next slide, please. So we had some significant challenges in getting this PRS pilot done. Apart from the usual of sort of setting up a study with a new consortium, there was obviously the headwind of COVID and whatnot. Nevertheless, I think we set out to test whether or not a transethnic. First of all, to test whether or not you could sort of do a federated PRS approach without sharing any of the primary data, and we can do that. It actually worked out pretty well, I think, after working out a few niggles. And then looking at the transethnic versus the population specific, I think this actually is pretty encouraging. We observed, pretty for the BMI, we observed a pretty decent score in the Shanghai cohort versus, for example, the large NHS1 study. So the type 2 was a little bit underpowered. I'm not sure that we can really use that to draw many conclusions, but the caveat being that the transethnic scores may be phenotype specific, and that we may have to take into consideration, the fact that local architecture in different diseases may not translate transethnically. I think we would benefit from having some more additional population-specific LD files, such as South Asians, and if there are other large groups that are joining, we may need to generate some more LD files for this sort of an approach. I would say that the data sharing agreement would facilitate work greatly. I think having that in place would make this project and others not only more easy to do, but you can then also sort of standardize the results and meta-analyze across those, and you can infer more from them. There are some new tools being released. So there's an LDPRED2 tool that's just been released in arXiv. And next slide, please. I'd just like to sort of acknowledge the people at different sites that really sort of stepped up and got us the data. Alex Pereira from Brazil, Bram Prinz from the UK blood donor cohort, Chris Flatley from MOBA, Yurong Long and Yaohua Yang from the Shanghai Men's, Pete Kraft from Nurses. And although they didn't provide any data to this, I'd like to thank Chin Huang and Hilary Martin for sort of volunteering to help us get some more LD files going forwards for the South Asians. And couldn't have done this without John Connolly. He did all of the sort of coordination, and Jonathan Bradfield and Xiao Chang from CHOP, who generated some of the scores for us. So thank you.
Thanks, Patrick. Wonderful presentation. And I'd like to echo Thomas Keen's comments from the data integration group that after so long discussing science and the planned projects, it's fantastic to see some results. And thank you particularly to you for all your hard work. I know results were being generated as recently as yesterday on this. And I know it's been a race to get things together for this summit. But really gratifying to actually see cohorts from IHCC working together in this federated data model and doing some great science. So, in terms of the next steps, I guess the immediate next steps for you then are to expand to some of the other cohorts from IHCC and try and include some different ethnic groups. Is that the main focus next, Patrick?
I would think so, yeah. I mean, I think we have a decent basis here, and I think we have some interesting preliminary results, and so it would be great to test those in larger numbers and in expanded populations and in different populations. I think it's actually from a tough point. It's actually we've actually got some interesting results and some hypotheses that could be tested here. So I think we should definitely carry on with this project and expand it further.
Great. And as you say, we've identified from that some of the challenges and some of the opportunities as well. And I think that can mostly feed back to the data integration and harmonization team as well about how we do this, as well as the processes for making individual level data shareable to make these kind of analyses easier in future.
Yeah, so I think that in the last session there was a presentation by Christina Young who was presenting the phenotype catalog. I mean, that sort of thing would be very useful when you're going in and designing these sorts of studies to see who has numbers across the different phenotypes. So, yeah, I'm sure as things come together, it'll make these sorts of projects much more easy.
Great. I'm conscious of the time. We don't have much time left in this session, but a couple of quick slides then on some of the next steps for the scientific strategy and enhancements work group, and then we can open up for discussion. I see there are already some questions on the chat function. So we heard this morning about the opportunities that IHCC might think about putting together around COVID-19, and that will be a whole theme in itself. But more broadly, we've been thinking about how we can better delineate the research focus within IHCC. How Can presented some of the ideas that were discussed at the Iceland summit last year. And there was some incredibly useful discussion at the end of that summit about how we better collate ideas from the group, how we prioritize ideas, And we can unpack that a bit more in a minute. We've seen about identifying cohort assets, what cohorts have already and how we can visualize that, but also what cohorts need as well. So how do we identify what are the gaps that we want to plug? And of course, how do we collaboratively pursue funding to address some of these questions? So in terms of the research focus, we're constantly thinking about ways we can identify projects to capitalize on what IHCC's distinct strengths are. How can we go beyond what we do on our individual cohorts? But particularly, we wanted to broaden the initial focus that we saw in some of the project ideas. So thinking about addressing areas beyond genetics, genomics, other omics, and how can we give a broader profile of projects that speak to some of the data that are collected in these cohorts. And so some of the initial exemplars we heard about previously were things about identifying risk factors for less common diseases where we need these extremely large cohorts. Thinking about other heterogeneous risk factors, not just genomic data, but what are the other lifestyle or other kinds of factors that are measured in these cohorts that might benefit from being studied across the world. And how about going beyond one disease at a time, thinking about multi-disease etiology. Those are initial ideas, but of course we're welcome and we'll try to collate further ideas, particularly to strengthen on the non-genomic side. As part of that, as Håkan mentioned, we already, prior to the summit, distributed a survey asking for cohorts to provide their some questions, some research priorities and interests. And the plan had been to use the Santiago meeting to take those surveys, to come up with several themes, to have breakout sessions where we could discuss within groups each of the themes we were interested in and come up with a set of priority questions. Clearly, that's been more challenging to do in a remote format like this, so we will need to regather our thoughts after this summit and probably send that survey out again. Thanks to those of you who already sent in ideas, but I think we'll need to put that out again so that we can collate further ideas and that we can design the highest priority questions going forward, thinking carefully about the kind of principles that Håkan mentioned earlier in terms of which projects we select. As you saw from the data atlas, we're already starting to do the job of collating the information about what cohorts have already measured, both in terms of omics and non-omics. And that will also help us then with identifying where are the gaps and also what could a project look like that aims to enhance cohorts, particularly in the low and middle income countries. So we heard earlier from Mary and Håkan that the Wellcome Trust have come forward with, we put a proposal into the Wellcome Trust to fund some more of these pilot projects. So we've heard about the Polygenic Score project, for which seed funding was given from the NIH. But there's now funding from the Wellcome Trust to kick off a series of pilot projects to try and replicate what's been done with the polygenic scores, but with a broader set of scientific projects. And so, as we just mentioned, again, these are short timeline projects with some initial pilot funding to get things going. Ideally, this will have to be projects that can work in the federated way that you heard about from Patrick, as we're not there yet with the bringing all the harmonized individual level data together. And as noted here, this is something that the executive team of the IHCC will round back on after the meeting to work out what should be the revised process for coming up with the best scientific projects to put forward for that pilot funding after the meeting, given that we can't discuss these easily face-to-face. And finally, now we have the proof of concept, we have infrastructure being built. How do we expand on the activities that we have been doing in terms of reaching out to funders? So you heard from Håkan that we'll hear tomorrow from a particular funder with an interest in Alzheimer's disease. How do we build on this? How do we find other disease-specific or perhaps exposure-specific funders who might be interested? Which other major funders, whether part of the HEROES or other organizations, might be interested to fund some of the high-priority scientific projects that we can put forward, and how do we go out and lobby for these? And then particularly on the cohort enhancement side, where we think about measurements and assays, continuing the conversations that have been had with industry, we'll have the session tomorrow about industry engagement, but also with platform makers and assay manufacturers. How do we demonstrate to them the value of IHCC and working closely together? So that's the other major area that the scientific team's been working on. The questions. So Eric, I'm hoping you've been keeping an eye on the chat and the Q&A.
I sure have. Yeah. So the first question comes from Ricardo Verduga. It seems that whether the transethnic scores outperforms the population-specific scores depends on sample size. European studies are largest. Would this conclusion hold if controlling differences in sample size across ethnicities? I think that's probably for Patrick.
Yeah, I think that's a good question. The best that I can do is point to at the Shanghai study. There they had decent sizes, and the transethnic score did outperform the population-specific one. It's not just that. There's also how large was the initial study for the Asian population-specific data, and so there are multiple different factors that we need to control for. I think if we had more data, we could better answer that question.
Okay, thank you. And Terry, I think that echoes the question that you raised in the chat. There's another point here about the very low number of diabetics in the MOVA study.
The number was very small. Yeah, I mean, they were. I would not necessarily, sort of use those results, but they were provided by MOVA, and so we included them. But yeah, they're too small. Yeah, for sure.
I mean, 27 can't be right. There's no way out of 28,000. But actually, I was asking a slightly different question, Eric, in terms of the transethnic scores. Have other studies tried to do this, look at transethnic versus population specific, and have they also seen the transethnic to perform better?
Terry, I haven't seen any studies that have necessarily addressed this question, and
I don't think there is any publication, Terry, on that. There have been sort of somewhat mixed population studies, but very, very limited. Mm-hmm. So there's nothing really. I mean, I think we will be the first, and that could be a driving publication, for example, in this area.
Yep, agreed.
And that was this is Jeff. That was kind of my comment in the chat, was about trying to understand what you guys think is the path to a strong publishable unit that demonstrates not only demonstrates what we could do as a consortium or as an initial proof of principle. So that's number one, and number two would be if you could highlight some of the challenges that we need to address to make sure that the next series of scientific forays in this direction are things that we can maybe accomplish more expeditiously.
Yeah, it's a great point. And if Decode can put together a New England publication in 12 days, we should be able to do it in 10.
So, what will it take to actually get this? New England would be nice, but at least. It obviously. I mean, there are several other cohorts that show that sort of are probably on the edge of getting us the data. I mean, having a little bit more data would obviously be nice. To sort of, drive the first publication. But I mean, these data, Patrick, they look actually quite nice. I mean, I think the Norwegian diabetes sort of, attenuated a little bit the impact on type 2 diabetes, but otherwise it looks good.
And the type 2 numbers are small across the board, apart from the Nurses' Health study. I don't know. I mean, this is what I was sent by Chris Flatley, so I don't know if that represents their true caseload or not. I mean, these nurses were presumably recruited at a younger age, so maybe that explains it. I think the path forward, if we got 23andMe on board with 500,000 samples and a bunch of phenotypes, that would actually help us out a lot. Would get us to towards a publishable paper. And if any of the other, larger cohorts jumped in, that would also get us there. We just need to have a little bit more data, I think, around some of the, the non-traditionally sort of tested populations like Northern Europeans and even African Americans. If we can get into South Asians, so if we can get the East London study on board, that would be a very interesting data set to have.
And perhaps the discussions we heard earlier about the charter and the policies and getting those in place will then perhaps free up some other cohorts who may have been currently unable to participate, who might be more enthused and freer to share their results as well would be good. But I think thinking going forwards about how we can maximize cohort participation in further pilot projects would be good, and whether there's lessons we can take forward from this initial experience to ensure we can engage as many cohorts as possible for future projects would really help.
Okay. Well, thank you, everyone. And as expected, the discussion has taken up nearly all of the time, which is as it should be. So I will, to respect everyone's time and get us off of this call and onto the next things on our agenda, keep the summary brief here. And so the long list of bullets and outcomes that came from both the Data Atlas and Infrastructure group earlier in the session, as well as the Scientific Strategy group, I will grossly oversimplify and just say that it's clear that we have proof of concept in both areas that these things can be done. And that there's the beginnings of infrastructure for how to do them, as well as lessons learned already coming out for how to improve them going forward, and that what we will see over the coming months, not just a year, but really active groups getting together and feedback being sought, is to flesh out what those initial proofs of concepts are, test them in some different ways, and build the plans to make them public, either through publications of the scientific studies, or again, getting the atlas out there after it's been appropriately vetted so that others can see what the power is of what IHCC can offer in terms of bringing things together and let them know things that we're finding and working on. Also, there are some action items for the cohorts that will be coming. I think the shortest term ones, again, in the simplified way, will have to do with really taking the issues for prioritization that the scientific strategy team has developed for the various phases of the work that's going on now that there is some seed funding to get several more projects underway, and providing input from the cohorts on what those opportunities are and where the gaps are, where IHCC, again, can contribute in its unique way to pushing this forward, and again, continuing to work on how to work together, which will also have iterative development as policy issues can be addressed by, frankly, the cohorts pushing the issues by trying to do this, and therefore providing concrete examples where we can look at that. So seeing the other work streams coming in and starting to really actively engage and collaborate with these two work streams going forward to help, again, flesh out that infrastructure. For the more medium term, I think that's where you all will start to hear from the Data Atlas and Infrastructure group to get feedback and test drive the Atlas once the initial validation has been done by the smaller group that they talked about, having tested, and then make sure that you're really giving them input on case studies. So please look for those opportunities to do that and be very active and vigorous in providing the responses. I think that's going to just make the tools that much better. So with that, we'll close and we'll continue to go forward. I'm really excited by the things that we heard today. I can tell from the discussions, both in the chat and things that people have volunteered online, that everyone is excited to finally be to the point of doing the science and having the tools to start playing with. So I will close and ask Jeff if there's anything else that you'd like to say to close out the day before we release everyone.
Thank you, Laura. Thanks for sharing this this session. It's been a great day, 6 or 7 hours, whatever it was that we spent together. Tomorrow we have an equally exciting opportunity. We have two distinguished keynote speakers, as you probably see from the agenda. Soumya Swaminathan from the World Health Organization that will also, she'll also talk about the WHO roadmap for COVID-19 as part of her remarks. And at the end of the day, Jeremy Farrar from the Wellcome Trust will be a sort of a closing keynote, not quite at the end of the day, but very close to the end of the day. And then we have a very robust scientific agenda. Many of the esteemed scientists that are contributing to the IHCC and were part of the discussion today will be presenting tomorrow. So I'm hoping that we'll have as strong attendance and engagement tomorrow as we did today. And thank you all for being here in this first virtual meeting, and wish you all a good day, a good afternoon, or a good evening, depending on where you are. Thank you.